Okay, it looks like all the usual people are here, so let's go ahead and start. So just a reminder, since this is exam week, there's another review session tonight. Don't forget, it's in the teaching lab from 5:30 to 6:30. So we left off talking about some introductory material to bacteriophage. And so just kind of keeping with that before we launch into really drilling down on one particular bacteriophage, lambda. Let's do a little bit of definitions. So some of these you probably know because I think we've touched on these in another section. But in a typical viral infectious cycle, as would occur most frequently in the laboratory, a temperate bacteriophage like lambda has its choice of either undergoing a lytic cycle or what's referred to as a lysogenic cycle. In the laboratory, for the most part, the lytic cycle is going to be favored because the bacteria host is very healthy, has lots of, is well provisioned with lots of resources. And so the genome circularizes upon infection of the bacterium, and viral gene expression is created. New little baby viruses are produced, and the cell lyses. Now, under specific conditions of the host, when this infection occurs, you can force lambda to make a different choice. Rather than to choose to make viruses now, if it finds itself in a situation where either the host cell is poorly provisioned, so it doesn't have a lot of energy, ATP, resources and such, it will, instead of going into the lytic cycle, will wait out the hard times. So it will adopt a genetic parasite type of a strategy where it inserts itself as a prophage into the genome and then just becomes another locus on the genome. So that bacterium, that lysogen, that's the definition of a lysogen at this point, is a bacterial strain with a prophage in its genome, can simply replicate every time the bacterium replicates. And this can go on for generations and generations. But at some frequency, something can trigger this bacterial cell, this lysogen, to have its virus resurrect itself from the chromosome to try and produce viruses to lyse the cell. The signal for this is usually the same type of a signal that would indicate that the bacterium may die. So the virus, this is a virus adaptation to make sure that the virus prophage doesn't perish along with the cell. So just by a couple of definitions, the prophage and the lysogen are here. So under most circumstances, most of the prophage genes are silent. They're not expressed within the lysogen. So shown here, this prophage, for the most part, 99% of those genes are going to be transcribed not at all or very low levels. But notice the emphasis on the word most. There is, and we'll talk about this at some depth, there is at least one viral protein that must be expressed from that lysogen, from that prophage, in order to maintain lysogeny. This process whereby the bacteriophage is incorporated into the genome and kept there is actually an active process driven by that prophage. So there is one gene that is actively transcribed and translated into protein anytime you have a lysogen like this, at least one protein. The rest of the genes are largely unexpressed, maybe very low levels, but for the most part you can think of them as being not expressed at all. So what kills the lysogen upon induction, or what kills the bacteria, is not simply the presence or even the replication of that viral genome. It's the expression of the virus genes, other than that one gene that maintains lysogeny. If those other genes become expressed, that's what's actually going to burst the cell. That's what's actually going to kill the bacterial host. In the lysogen, that viral gene that I mentioned that has to be expressed is a repressor protein. So this is a protein made in the host. It's a viral gene, but its protein product serves to stop the expression of virtually all viral genes. So it's just keeping everybody quiet. Because remember that some of these virus genes, as you can imagine, are very toxic to the bacterial cell. There are genes encoded within these prophage genomes that literally will poke holes in the membrane of the cell and cause it to depolarize. There are other enzymes that produce lysozyme, couldn't come up with that word today, lysozyme, which will degrade the peptidoglycan. So it's very important for the virus's continued survival that it maintains tight control over gene expression during this lysogen phase. Once the cell becomes induced, however, if something such as ultraviolet light or perhaps, bless you, an antibiotic treatment comes into play, then DNA damage and stress to the cell can cause that virus to now lose control of its gene expression. So that repressor protein will no longer be able to mediate control. And so the virus will resurrect itself from the genome and go into the lytic cycle. But once that lysogen loses control of the viral gene expression, the virions are going to be produced and the lytic phase of the life cycle begins, and there's no hope anymore for the bacterium. That cell is now absolutely dedicated to the lytic cycle. It's not going to go back into the lysogen. And here's the other odd thing that we don't really understand that well, that in any population, even in the laboratory, of a bacterial lysogen, spontaneously anywhere from 1/10 to 1/10,000th of a percentage of the cells, for reasons that no one understands, will spontaneously lose control of lysogeny. They will stop producing that repressor protein and now enter into the lytic cycle, and those cells will be lysed and release viruses into the environment. Now, like I said, we don't really understand what causes that. It could simply just be a random stochastic event that really doesn't have an induction. It could be something like a cosmic ray or an unknown ultraviolet photon somehow hits the DNA and damages it. But in any population, there's always some cells that are producing viruses. And if you put the bacteria under more adverse conditions, such as high levels of ultraviolet light or antibiotics, then the percentage rises. The cells that become overly stressed, and so the virus decides to get out of town before the host gives up. But what's interesting about this is that here's a population of lysogenic bacteria with a prophage in it. 1 out of, say, 10,000 of these cells will now enter the lytic cycle and produce virions, releasing them into the environment, which are completely infectious virions. They have the capability of infecting and lysing another bacterium. Yet, they don't infect the other cells that have the prophage in them. So the lysogens are actually immune to superinfection. They cannot be infected by that same virus again. This is a process very adequately known as immunity. So this is actually due to 2 reasons. One is that repressor protein that we talked about that's holding the lysogen in quiescence, or at least holding the viral gene expression in quiescence, that repressor protein does 2 things. First of all, it represses the expression of viral genes maintaining lysogeny. We've already talked about that. But also, very importantly, is that repressor protein that's being produced in the lysogen can act in trans. It can act on incoming viruses, and that's exactly what it does. So that level of repressor protein that's being synthesized in that lysogen is not only making gene expression of the prophage quiescent. If other identical genomes come in, that protein has the ability to silence the gene expression of the new viruses coming in. This is what gives immunity to superinfection in these lysogens. And again, it all comes down to the function of this repressor protein. So conditions that induce the lysogens to enter that lytic phase typically destroy the phage repressor or halt the synthesis of the mRNA. This is going to sound somewhat familiar, and we'll go through the details of this specifically with lambda. But you've already kind of seen this process before. This is very reminiscent of the SOS response. Remember that when conditions for the SOS response occurred, DNA damage, you had the destruction of a repressor protein, LEX-A. You're going to see the exact same thing in E. coli and in lambda. That repressor protein that's holding everybody down in terms of their gene expression is going to be destroyed by the same process. When the SOS response is turned on by the bacterium in response to a potentially lethal stress, it's as if the virus is eavesdropping on that conversation. So the bacterium says, okay, we have some DNA damage, let's start breaking down LEX-A to let loose these DNA repair mechanisms. But at the same time, the virus listens in on that conversation and says, oh no, we're also going to allow that REC-A protease to degrade the repressor protein. So that now, in addition to inducing the expression of DNA repair mechanisms, you're also inducing the expression of all of the viral genes for that virus. And so the virus will now resurrect itself, and from the virus's point of view, hopefully lyse that cell before the cell actually dies. Okay, so that's just some general information, and now we really want to apply some of that to the one specific paradigm that's best understood in terms of temperate bacteriophage, and that's lambda. So this is an electron micrograph of lambda. So you can see some similarities structurally with viruses that we've talked about before, the T4 and T7. They have these icosahedral heads, which contain the double-stranded DNA genome. They have this kind of striated or helical tail. This tail is, however, not contractile, and it doesn't have the 6 tail fibers that things like T4 and T7 have. It has, and you can see it just barely in these micrographs, a single tail fiber at the end, which accomplishes the same thing as those 6 tail fibers on the lunar lander type T4 bacteriophage or T7, but it has only a single one. Now, that lambda virion that I just showed you in the electron micrograph is going to initiate infection with E. coli by binding to a specific receptor. And this is a pretty ingenious specific receptor, because we've touched on the fact several times in this class that maltose is very much a favored substrate of E. coli. It loves that glucose-glucose dimer, which is broken down in the environment from things like cellulose or starch. It loves to transport those things in. It's a major source of energy for E. coli. So what lambda has done is it's taken advantage of the fact that E. coli really needs to express an outer membrane protein that allows that maltose to get into the periplasm. That protein is called LAMB. That's the maltose transporter through the outer membrane. That's actually the protein that the lambda tail fiber is specific for. So it'll bind to that LAMB and then inject its linear double-stranded genome into the cytoplasm of that cell. That lambda genome that's packaged in that icosahedral head is linear. It's a double-stranded piece of DNA with single-strand overhangs at the end, but it's a linear piece of DNA. But almost immediately upon injection into the cytoplasm, it takes on this structure here shown, this circular structure. And here in this purple box are what are known as the COS sites, or cohesive ends. Those are single-strand overhangs of about exactly 12 nucleotides that are complementary to one another. So by simple base pairing, these two cohesive ends from an otherwise what had previously been a linear piece of DNA have circularized to become this circle. So this is again showing it here. Here is the DNA in the form that would be packaged within the icosahedral head of lambda. And these are the single-strand overhangs, the COS sites. The base pairs here on the right-hand end are complementary to the left-hand end, so that upon infection, you can get a circularization. This figure also kind of nicely shows some of the genes that we're going to go through in terms of their function. And so you can see that this virus, and this is actually a rule for many viruses, actually have their genes oftentimes clustered based on their function or based on the timing with which they're going to be infected. or going to be useful. So when you look at some of the genes that are involved in lysogeny, you see them here in red. Genes in lysis are clustered here. Genes involved in recombination. But at this point, we're just really talking about the fact that this genome circularizes. And that ends up being very important in terms of how it replicates. We don't take the time any longer in this class, because I've tended to shorten up this section of the class, to go through the replication of T4 and T7, but those are linear genomes that never circularize. So they have to have a very unusual mechanism in order to replicate their genomes, because linear genomes are kind of problematic when it comes to replication. So these types of genomes, the linear genomes, tend to form what are known as concatamers. Lambda gets around that by doing this very nice circularization, and Lambda is not alone. I mean, this is a very common strategy that's used by temperate bacteriophages. So typically in the laboratory, at least, lambda infection almost always results in a lytic pathway. So we've covered this a little bit before already, but lysogeny occurs typically in response to specific environmental conditions that lambda detects upon infection of that cell. Not before the infection, but after injecting its genomic DNA into the E. coli, it then knows whether that cell is healthy or sick. But at that point, if it's sick, it can't do anything about it. The die is cast, it's stuck there. So we've talked about this already. The nutritional starvation of the host is one thing that triggers lysogeny. But the other very important environmental condition that we haven't touched on is what's known as high MOI, or high multiplicity of infection. If an E. coli cell is infected by, say, 50 or 100 lambda genomes simultaneously, even if that E. coli cell is fat and happy and has all kinds of energy and glucose and just everything that a hungry little virus could want, under high multiplicity of infection, what happens is that the virus will actually not go into lytic phase, but instead go into the lysogenic phase. The reason for this is a little bit, well, it's not hard to follow, but it's not something that's inherently, I think, logical. But what happens is that this is an adaptation of the virus to make certain that it doesn't kill every host in the environment. If the conditions are such that any one bacterium is going to be infected with 50 or 100 lambda, that's an indication that there is a danger to the virus of lysing every potential host in that environment. So if every host goes through lysis and produces viruses in the environment, and there are no new hosts to infect, those viruses are just sitting there, doing nothing, which doesn't seem like a bad thing. But remember, one of the tenets of viruses is that they don't have a metabolism. They don't have the ability to repair their DNA, or if it's an RNA virus, to repair their RNA. So anything that damages their protein structure, like a protease, anything that damages their nucleic acid, like ultraviolet light or cosmic radiation or toxins, is irreparable. So viruses like these don't hang out in the environment for tremendously long times. They're not tremendously stable. So the virus has adapted this ability to recognize when it may actually eat itself out of house and home and kill every possible host, and thereby just be a sitting duck for any stray photon of ultraviolet light or any protease that might nibble a little bit off of one of the capsid proteins and damage it. So it tends to keep hosts alive by going into this lysogeny period. It's a very clever adaptation that the viruses have gone through. Now, lytic bacteriophage, or I should say, bacteriophage like lambda that are temperate, that can be lytic or lysogenic, will oftentimes produce what are known as cloudy plaques. This is not a good picture of it. These are plaques on a lawn of bacteria on a plate. So you're used to looking in the laboratory at colonies growing on a plate. When we grow viruses on plates like this, we put a lawn of bacteria on there, and then we put on smaller numbers of viruses. So each one of these little clear spots in an otherwise uniform field of bacteria is like a little nuclear explosion that's gone on here, where all of the cells have been lysed by the virus, and the virus has diffused out and formed this plaque. But you can actually see a different phenotype in the plaques because the temperate bacteriophage, remember, these are viruses that at some frequency can cause their host cells to become lysogens and therefore be immune to being lysed. When that happens, you get surviving bacteria in that plaque. And so that gives you a cloudy appearance. You can't look through that plaque very clearly, as opposed to a virus like T4 or T7, for instance, that has no ability to make a lysogen. It lyses the cells or nothing. In that particular case, there are no cells left to diffract light. So those plaques are very clear. So you can sometimes tell something about the phenotype of viruses by plaquing them on a bacterial lawn like this and seeing differences in the plaques. So just as an example of this is so I used to work with a virus so Dan Crystal and I used to work on a bacterium that degrades feathers in birds. And so as a offshoot to that, I started working with viruses that infect that bacterium that infects birds. So this is an uninoculated plate. I'm just holding up to my cell phone. And so you can look through that plate. There's no bacteria growing on it. If we grow the bacterium Bacillus licheniformis as a lawn, you can see now that plate is no longer capable of, you just have a lawn of bacteria. And what I had put on here before, or actually a student, Ariel, had put on here before, was a virus that she had isolated from a sparrow that had this bacteria phage in it. And you can see now these plaques, these areas where the bacteria have been completely lysed. This is a lytic bacteriophage. It has no capability of entering into lysogeny. So when it infects these cells, it just kills them all. It just wipes everything out. As opposed to, this doesn't project very well, this is another virus that we isolated from this, which produced turbid plaques. This is a temperate bacteriophage. So some of the cells in here have been lysed. So light goes through here a little bit better, but you can't read through this plaque any longer because there are a number of lysogens that have been formed in this. So temperate bacteriophage tend to form these turbid plaques like this, as opposed to lytic bacteriophage, which tend to make the clear plaques. But anyway, back to lambda then. So lambda, the messenger RNA that's produced upon infection, really depends on this circularized form. This is where lambda genomes begin to be expressed. So the first gene expression, or matter of fact, I should say, all of the lambda gene expression is actually done by E. coli's RNA polymerase. We don't have time to go through other viruses, but other bacteriophages sometimes bring in their own RNA polymerase or their own sigma factor. Lambda just uses the plain old RNA polymerase with sigma 70 that E. coli has. But it begins to express the lambda genome at particularly two promoters called promoter left, P sub L, and promoter right, P sub R. So in this diagram here, you can see P sub R here in this yellow box. So this yellow box is where we're going to spend a lot of time over the next lecture or so, because this yellow box is the area that lambda uses to make the decision whether to go into the lytic life cycle or into the temperate or lysogenic life cycle. So the first genes that are going to be expressed, whether the bacterium goes into lysis or lysogeny, are going to start from transcripts coming from promoter right. So that would be this transcript here called R1, which is not as monolithic as that might look. I'll show you later that this transcript shown here as R1 is actually multiple transcripts of different lengths. And then there's promoter left, P sub L, which drives transcription of a transcript shown here as well, called the L1 transcript, which again is a little bit more complex than it's shown here. But these transcripts, as I mentioned, are not monolithic as they're shown here. I've added some little stars here, these red stars, to indicate factor-independent terminators. Again, exactly what we've talked about in another section of the class. These stem loop structures followed by a poly-U tail. There are DNA structures here, here, and here, which function to stop RNA polymerase. So the first transcript that's going to be made from promoter right is going to proceed through operator right, that's what OR is, and this very important gene called CRO, And then for the most part, if not entirely, it's going to stop at that transcription. So this transcript that's shown here as R1 going all the way down through the gene Q initially is not made in that full-length version. Instead, all of the transcripts will stop at this first terminator molecule, or terminator region, I should say. Same thing is occurring from promoter left. E. coli's RNA polymerase with sigma 70 recognizes the -10 and -35 of promoter left very well and begins to transcribe through operator left, O sub L, transcribes through this very important gene called N, and then stops at this transcriptional terminator. So this sets up a situation where the only two genes that are being expressed in the earliest part of the lambda infection are CRO and N. Let's first deal with the N protein. The N protein is a fascinating protein. It's actually an anti-terminator. This is a little different than the anti-terminator that we talked about with attenuation. That was an RNA structure. This is actually a protein which allows RNA polymerase to actually transcribe through through those terminators from promoter left and promoter right. So when this first protein, or first whispers of this protein N are produced, this transcript coming from promoter right is no longer going to stop at this transcriptional terminator. It's going to continue on to transcribe through genes C2, O, P, and then it hits another terminator that it doesn't do a very good job with, but it's not entirely bumfuzzled by this terminator. More often than not, the transcription will stop after the gene P. But at some frequency, maybe 1 out of 10, maybe 1 out of 20 times, that N, anti-terminator, allows the RNA polymerase to transcribe through the protein Q. So now, and I'll show you this in a slide diagrammatically, but think of it this way. This transcript that's shown here from the textbook as R1 is actually 3 different transcripts of 3 different lengths. One goes through CRO and stops. Another one goes through CRO, C2, O, and P and stops. And then the third form is this full-length one that's represented in the cartoon here that goes all the way through Q. What this does is it sets up a situation where the protein product of Q accumulates very slowly. Q is actually a protein that's absolutely crucial for the expression of this transcript shown here as R2. Notice the genes, the function of the genes that are contained within the transcript R2. R and S are the gene products that will lyse the bacterium. S puts a hole in the membrane. R is a lysozyme. So if those genes are expressed, the bacterium is going to die. But you don't want to kill the bacterium too quickly because at this point we haven't expressed all of the virus genome. So Q, the protein which is an anti-terminator in fact, for this R2 transcript needs to build up slowly. And it does because it's present on only that rare R1 transcript that makes it through that second terminator. But when Q does eventually over time, I mean now we're talking like 20 minutes maybe, not long periods of time, but if Q builds up, it will now allow the transcript of these lysis genes as well as all the structural proteins that make up that icosahedral head as well as the tail. So now you can have assembly of the virus late in the infection, all based on the fact that that Q protein accumulates slowly. This is just an image that I found from online that kind of illustrates a little bit about how this N protein, this fascinating anti-terminator protein, functions. So here is, exactly as you remember it, what a factor-independent terminator, transcription terminator looks like. Here's RNA polymerase shown in the box. This gold strand is the nascent RNA that's being synthesized. And here you have the stem loop that's formed, and this would be the poly-U tail. So this would be an inherently very unstable structure, and so this would tend to have RNA polymerase fall off at that point. The N protein, this is why I think it's so fascinating, because N protein does not function by itself. Here's the N protein. This is the anti-terminator produced by lambda. it interacts with several other proteins. These are not virus proteins. These are not proteins that are normally even involved in transcription. This N protein, this anti-terminator protein, has this really weird ability to recruit completely unrelated proteins. They're not even involved in transcription, yet it puts them together in this complex, which binds the RNA as it's coming off from RNA polymerase, and it's basically like pulling on that RNA to stop the stem loop from being formed. So because the stem loop doesn't form, because of tensile pressures, then the RNA-DNA heteroduplex continues relatively intact. And so RNA polymerase doesn't stop at that terminator because N has formed a collaboration with unrelated proteins that it's never met before to do this job. So this is how anti-terminator proteins work. And okay, so that N protein, as I mentioned, I've gotten ahead of my bullets again, is not as effective an anti-terminator before the Q gene. So that allows for the concentration of the Q protein to build up very slowly. And that's really important because once that concentration of Q builds up to too high a level, then the R2 transcript has no choice but to be transcribed, and that cell is going to go through lysis. But if the cell needs to go under lysogeny, if the conditions are such that it would be beneficial to the virus to undergo lysogeny, this is not a good situation. So the decision still needs to be made. The decision really to go lysis or lysogeny needs to occur before this Q protein builds up to a high enough level to induce the transcription of the R2 lysis. Once that occurs, the cell is just dedicated to lysis. There is no turning back at that point. Now at this point, up until this point, we're still haven't made a commitment to lysis or lysogeny. But at this particular point, the lambda genome has been replicating by a theta type replication. So just like we talked about with plasmid replication. And you actually see the similarity between this and a plasmid. This circularized lambda genome actually has an origin of replication called ORI-V, which is just like an origin of replication in an F plasmid, for instance. And so by theta replication, this circular genome will turn into 2 circles. And those 2 circles will turn into 4 circles. So you've had this kind of duplication of the lambda genome occurring up until the point where Q builds up sufficiently. So at this point, you still have the possibility of making a decision. The virus still has the possibility of deciding between lysogeny and lysis. So let's first deal with the decision to go into lysis, because at least in the laboratory, this is the most common way that lambda is going to go. So we'll first talk about lysis. So as I mentioned, we're going to spend, let me back up for a second just to orient you. We're going to spend a lot of time in this yellow box here. Remember that O stands for operator, so you know what's going to happen at an operator. Repressor proteins are going to bind. Promoter, you know what binds there, RNA polymerase. And then you have this gene C1. This is a protein encoding gene. That region is actually crucial to the decision. This is where the decision is made. So if the cell is going to go through lysis, what's going to happen, and we'll go through the steps of this individually, but just to give you a general broad brush overview of what's going to force the bacterium virus interaction to lysis, is that this protein CRO, the first protein that's synthesized from promoter right, shown here, that CRO protein is a repressor protein that has the ability to bind to operator sequences. So this operator right actually has 3 different regions that can bind to that repressor protein. But these 3 DNA sequences, operator right region 3, 2, and 1, are similar, but they're not identical. So they all have the ability to bind to CRO, shown here as these red balls with C's in them. But operator right region 3 has the highest affinity. So when CRO proteins are expressed even at small levels, this is the first place that they're going to bind because they bind the best there. It's not until this site is occupied and CRO levels increase further that they'll bind to operator 2. And then eventually, if they build up to even higher levels, they'll bind to operator 1. But notice one of the things that happens, the first place that the CRO protein binds, this operator 3, the location of this doesn't affect promoter right. The promoter right, which is transcribing CRO, is unaffected. Transcription goes, matter of fact, it may even increase the transcription to promoter right. But notice that the only, so here's a promoter going in the other direction. Promoter right sub M. We'll talk about that promoter in a minute. But that's a promoter that has the ability to express this other protein, C1. This gene here makes a repressor protein as well. The whole battle, the whole decision between whether a cell becomes lytic or lysogenic depends on who wins this battle, CRO or C1. Anytime, so just as a way to remember some of these proteins, anytime you see in lambda a protein that has the lowercase c1, so there's actually C1, 2, and 3, those are proteins that are critical for lysogeny. They're not involved in lysis. As a matter of fact, the C stands for clear, because before people understood the genetics of this, they realized that there were mutants of lambda that would make a clear plaque instead of a turbid plaque, meaning that if you were making a clear plaque, that meant that virus had lost the ability to form a lysogen with the cell. It could only do lysis. And so they called these mutants C1 for clear 1, clear 2, or clear 3. We now know the genes that are responsible or where those mutations were. And so they bear the historical names C1, C2, and C3. But when you see those are the genes that make proteins that are crucial for lysogeny. But we're talking about lysis at this particular moment. So remember that the CRO protein will first bind to operator 3, which really functions to at least reduce the possibility that C1 can be expressed. So if CRO is going to win the battle, it needs to stop the transcription of C1. And that's what it does by binding first to operator 3. It tries to stop the transcription of C1. Now, here's perhaps, I don't know if this is a better figure or not, but I like this figure. So at the point where the concentration of Q is high enough to allow that transcription of that lytic transcript, R2, the CRO protein is at an even higher level. Because remember, that transcript coming from promoter right is actually in 3 different lengths. One length just has CRO, one length has CRO through O and P, and the other one through Q. But all 3 of those forms have CRO in them, encoded within them. So the CRO protein gets to very high levels. So if the bacterium has reached the point where there's enough Q to induce the transcription of that lysis transcript, the R2 transcript, then CRO is even higher level. So what's happened is that the CRO protein is a repressor binding to operators at the right-hand promoter and the left-hand promoter. And as I mentioned, these 3 sites are not bound equally by CRO. CRO binds first to operator 3. Notice that operator 3 lies right on top of this salmon-colored box with an arrow to the left. That's a promoter. So that's one of the promoters that can possibly drive the transcription of C1. C1, think of it as the mortal enemy of CRO. CRO gets a little bit of an upper hand at this point. It does its best to stop the transcription of C1. But it also binds to site C2 as levels get higher, and eventually it stops its own transcription. It has the ability to bind only at high levels to this lowest affinity site, site number 1, and this operator of the right. And notice that that operator is right on top of the promoter, the P sub R, the right-hand promoter. This salmon-colored box with an arrow oriented towards the right is what drives the transcription of CRO. But when CRO reaches too high a level, it shuts itself down. It shuts down all of the transcription to that level. It doesn't need any more. It's got enough Q to lyse the cells, so it shuts itself down. So that promoter to the right is only blocked when site 1 is blocked, and that only occurs when CRO concentrations are at very high levels. That promoter right and promoter left also eliminate the transcription of the C2 and C3 proteins. So again, remember, when you see the lowercase c gene, that's a gene whose protein product is important for lysogeny. So CRO is not only shutting down, as best it can, C1, it's shutting down C2 and C3. Those are proteins that we'll talk about later that actually help to promote the transcription of this C1 gene. So this is another way to look at it. Maybe this is an even better picture that I found recently. So here is, this actually, instead of saying N is further down, think of the C1 protein as being here instead of N. I probably should have changed that. But here's promoter right for the maintenance of lysogeny. That's what M stands for. This is the promoter that would, if it could, drive the transcription of the C1 protein and push the system towards lysogeny. Here's promoter right that drives the transcription of CRO and all of those other genes downstream from it. Here's its minus 35 site, here's its minus 10, and then here in these pink boxes are the 3 operator sites. And you see the inverted repeats indicating that, remember that repressor proteins, DNA binding proteins, tend frequently to bind to inverted repeats. And so these operator sequences have inverted repeats. And so when CRO binds first to this operator 3 site, it's right on top of the promoter that would drive the transcription of C1. So it's a perfect repressor protein. It tries its best to stop the expression of C1. CRO builds up to a higher level, it binds to operator 2, which probably doesn't have a major effect. It might actually stimulate the infection, but you can see that, or the transcription, but you can see that it doesn't really overlap heavily with either of the promoters. But then when CRO binds to, or gets to an even higher level, such that it can bind to this operator right 1 region, shown furthest to the right, that's going to stop the transcription of CRO. Here's just another way to look at this. You may find one of these, I put up multiple pictures just hoping that, you know, what resonates with me might not be the same as what resonates with you. So as you look through these pictures, there's a certain degree of redundancy. But this is a fairly complex process, I recognize that. And so hopefully one of these kind of sets you going on the right direction. But here's the C3 protein gene. So we're now talking about the promoter to the left. So that promoter to the left also has operator sites that can be bound by CRO. So CRO first binds here and tries to stop the transcription of C3. So C3 is a protein that, if it's made and if it's maintained, will push the cell towards lysogeny and not towards lysis. So again, just to show this in a bigger picture, remember that this R1 transcript is actually 3 different sized transcripts. All 3 of the transcripts encode CRO, so you can see how CRO is going to build up very fast to very high levels, whereas Q is going to develop its concentration very slowly over time, and that's very important. So now, up until the point where up until the point, what is my point? Oh yeah, up until the point where Q rises to a high enough level to induce the transcription of R2, this replication, the theta type replication, has been occurring based on the activity of these 2 genes, O and P. O and P are the genes that facilitate theta type replication. So as long as O and P are there and being made, 1 circle is going to turn into 2, 2 circles are going to turn into 4. But when CRO builds to a very high level and shuts down all of the transcript of R1, the O and P proteins disappear. They go away. They have a half-life. They're broken down pretty quickly. Now what happens is this allows the replication of the genome of lambda to switch. It's no longer going to replicate by it's no longer going to replicate by theta type replication. It's going to switch now to rolling circle replication. Because this is important because now the cell is going to lyse, right? It's going to produce little baby viruses. And remember the form of DNA that gets packaged into those icosahedral heads. It's linear. It's not circular. So continuing to make circle after circle is impossible to make new viruses because you cannot package circular DNA in a lambda head. It must be linear. So that's why there is this switch late in the infection from theta to rolling circle replication. And that's driven by the fact that these O and P proteins go away once that transcription stops. So it's going to switch into classic rolling circle replication, just like we talked about with the F plasmid. So again, here's another parallel between plasmids and viruses. This is a virus replicating in the same way that the F plasmid does. But now, rather than have that single strand piece of DNA that's being peeled off by rolling circle replication, rather than have that going through a conjugal pore into another E. coli cell, instead it's duplicated within that very same cell. So basically what happens is that you just make what are known as concatemers, long strings of double-stranded genomes of lambda, but linked one to another. So you can have these long strands that can have 20, 30, 40 different lambda genomes, but now all covalently linked together in a concatemer. So that theta-type replication driven by O and P generated numerous circular copies, and this helps to magnify the process, right? Because now rolling circle replication is occurring not from one circular lambda genome, but from 2 or 4. And then eventually these concatemers are going to be cleaved at a specific site by what's known as the lambda endonuclease. This is a restriction endonuclease that is encoded by the lambda genome that's going to cut and leave those 12 nucleotide overhanging sites that we started off talking about, those COS sites. So it's going to cut here, it's going to cut here, and it's going to leave those 12 base pair overhangs, but most importantly, it's going to generate a double-stranded linear piece of DNA that can then be packaged into the virus. So this ensures tremendous numbers of chromosomes are produced that can be packaged in their proper form, in the linear form. Okay, I think rather than push our way into lysogeny, I think I've given you enough alphabet soup for one day. I'm going to let that settle into your brains for a little while, and then on Wednesday, we'll talk about the lysogenic pathway. See you guys on Wednesday or tonight at the review session.